All right, well, welcome everyone to the biomanufacturing program at Laney College. Thank you for all the guests, community members and PDA members and others that are joining us today. Those of you from Skyline College, thank you for joining us. We love partnering with Skyline. Your professor, Nick Kapp, is a big supporter of ours, and we support you guys and everything you do. And thank you, Maria and other PDA folks for joining. So today I wanted to welcome Ethan Oxen from ABPDU, that's the Advanced Biofuels and Bioproducts Process Demonstration Unit. You can see a picture of it in the background there. Before we begin, and as I introduced Ethan, I just wanted to say that Ethan is a former Laney College student. He took chemistry at Laney College, and a little bit of a downer of a note here, but one of our longtime chemistry professors, Stephen Corlett, who taught organic chem for over 15 years at Laney, just passed away after a long battle with cancer. So I thought it would be appropriate for me to dedicate this this talk today to Stephen Corlett and his memory. Great. So Ethan Oxen, he's actually spoken to our program before last semester. He did a similar talk on upstream processing, and that was actually pre-COVID. And so we didn't actually have it recorded. And it was such an amazing talk and such an important topic for our program and what we teach in bioprocessing that I wanted him to come back so that we could all benefit from what it is that Ethan knows about upstream processing and what he does at ABPDU. And by the way, I'll just mention, although it's a little late now to apply, I'm sure Ethan will mention it, but the community college internship program offered through the DOE, which happens every semester, the application for this upcoming spring semester closes tomorrow. So those of you in my program, you've known about it for a while, but keep that in mind if you're looking for an opportunity to get some real hands-on experience at an amazing laboratory that partners with commercial industry. ABPDU has taken on several of our students as interns, and it's an amazing, it opens up a lot of doors when you train at a place like that. So you'll learn more about that today. So Ethan, welcome, and go ahead and take it away.
Great. Thanks for the kind introduction, Doug, and I'm looking forward to speaking to you all today. We'll have a lot of time for questions. If people have immediate kind of burning questions during the presentation, I'm okay being interrupted, but we can try to keep the flow pretty constant if you don't have any burning questions. So last time I gave this presentation, I think the topic was that Doug assigned me basically was yeast versus bacterial fermentation. So this time I brought a little bit of a more general presentation about fermentation process design and scale up, which I hope is relevant to you all. I think in the last presentation I also talked a lot about a lot of these concepts as well. So I didn't want to just stick to just compare and contrast. So I'm going to share my screen. Let's pull this up. Maybe someone can give me a thumbs up if they can see my screen. I know I don't have it in presenter mode yet, but okay, great. So let's see. Okay, are you seeing the slideshow? Okay, I've got some thumbs up. and Doug is even using the emoji thumbs up, so you get some extra points for that, Doug. So let's get started. So as Doug mentioned, my name is Ethan Oxen. I'm a process engineer at ABPDU. This is 5885 Hollis Street, which is where we're located. So this lab also contains Bayer, Amaris, and JBay, the Joint Bioenergy Institute, which is also part of LBL, which is the kind of larger lab that ABPDU is a part of. So let's get right into it. So let's start with the most basic thing. What is fermentation? And when we're talking about fermentation versus upstream process development, that can get a little bit confusing. I think in upstream, people could potentially, depending on the way you designed your process, you could also include like deconstruction of biomass and producing feedstock. If that's something that you guys are interested in the future, we do have people at our lab who work in that area, but that is not my expertise. So I'm not going to be talking about that today. So we're going to go with talking about fermentation process development. So the most basic question, what is fermentation? So there's two major kind of definitions of fermentation. So the first one is probably the better known by the public. It's an anaerobic biochemical process that produces energy, regenerates NAD, and is a reduced byproduct, right? So if you look on the left, this is probably the most famous fermentation pathway that people, even if they don't know the biochemistry, they maybe know this part. This is the process for producing beer from Wikipedia. And what's interesting as well, and some people may not realize or piece this together, is that our bodies also perform fermentation, right? We don't produce ethanol, we produce lactate, or if you've heard lactic acid when you get the burn from working out. So this is Usain Bolt winning by a landslide in the 100-meter. All of these folks are producing a lot of lactate because they're overloading the oxygen limited metabolism that they have. So that's just some basic kind of fun introduction. So what we're going to actually talk about today is this different kind of version or term for fermentation. So the use of bacterial, fungal, algal, mammalian, or insect cells to make useful products. Those can be drugs, chemicals, fuels, etc. Food. There's really a wide variety of different products that can be produced by fermentation. That's part of what makes it so exciting. So this is actually from my colleague, JP Prahl. I forgot to put the acknowledgement down here. If I have another slide, I also stole from him. So I changed it a little bit, but this is a kind of helpful place to start in terms of fermentation, in terms of the kind of dichotomy between fermentation that you see. So we have industrial biotech on the left, which is what we as a lab primarily deal with. We have produced some proteins that have medical applications. So we've kind of gone into the other side of things. We've done some cell culture before, but this is the left is kind of our bread and butter. So you're using microbes, bacteria, yeast, fungi, algae, low to mid value products, so fuels, chemicals. And then when you're talking about the scale that you're running these fermentations at, you're talking about hundreds of thousands of liters ideally. That can vary a little bit, and we'll talk about different forms of fermentation. So that's not a hard and fast rule, but this is just a helpful dichotomy for you to understand how to think about these differences. So you have days per campaign, so you're doing your campaigns pretty quickly. You need high productivity. We'll talk about that more later as well. You need a clean environment. I'm sure you all know the difference between clean versus sterile. And you need more flexibility. Or you have more flexibility because you don't have to adhere to the same kind of GMP standards that you do with a pharmaceutical process, right? Like once the pharmaceutical process is developed, you really need to stick to it. So this image, this shows the Novamont Genomatica 1,4-butane production plant, kind of a stand back picture of it, which is one of the larger production plants out there that uses a biocatalyst to produce a useful product, 1,4-butane, which can be used for polymer production. So on the right side, I'll mention it here because I think it is important, and you guys have learned about it as well, but this won't be the major focus of the presentation. It's cell culture and pharmaceutical biotech. So mammalian cells, insect cells, some microbes. I mean, most famously, you have E. coli for the first production of insulin by Genentech. So you definitely have some cases where you use microbes to produce, but for instance, for a lot of the new biologic drugs out there, they require glycosylation. And there's some efforts to use fungi and to use yeast to produce glycosylated proteins, but I haven't seen anything actually be commercialized in that space. So you kind of need to use mammalian cells so you can have that same glycosylation pattern and to produce an antibody. These are very high value products, right? So the process development kind of ethos of pharma versus industrial biotech is different for that reason. There's a lot more focus on making the most cost-effective process if you're making an industrial product like a fuel or a commodity chemical. Your competition is petrochemicals, which are very cheap. So you really do need to bring your costs down. I'm talking tens of thousands of liters from fermenter capacity, sometimes even less can be viable. Usually weeks per campaign. Some of them will be faster, but generally on a longer time scale because the cells, especially when they're using mammalian cells, because they just grow more slowly and they're more complex cells. You need an absolutely sterile environment, and then you have much more paperwork and documentation with GMP standards. So this is a Genentech bioreactor for cell culture at the South San Francisco location. So you can see the beautiful web of lines and steam and CIP that you can use to transfer in everything without it ever hitting the air, right? So you can just sterilely transfer all of your liquids in and out. So we're going to go over some common bioreactor types. There's going to be one, there's going to be four we're going to talk about. So one we're going to focus on, but I wanted to introduce you to some of these other ones. So we'll see if this video has sound. I'm going to try to mute it right when I start it. Okay, I think it's muted. Perfect. This is a wave bioreactor. I think this video is actually pretty cool. So this just shows, kind of in the video, you can watch it yourself if you want. This wave bioreactors, so you can see you can have your control pH and DO in these reactors. This is like, I think, a 500 liter wave reactor. These are really useful if you have shear sensitive cells to use in seed trains sometimes in pharmaceutical processes and even for the final product production. One of the other benefits, you can see there's this like kind of bag here. Or in small scale, you can see the bag is it's single use. So it's easy in the sense that they can sterilize these bags through usually, I believe, gamma radiation. So you can have the bags be completely sterile. You open them in a clean room, put everything in within that clean room, and you don't have to worry about having steam set up and sterilizing in other ways. So that's kind of a benefit of using the wave bioreactors. And it works well if you have mammalian cells or insect cells. It's where I've seen these used before. Not suitable for high density fermentation, kind of for obvious reasons, but you know, you're not going to be able to grow up in a coli high density culture in one of these bags, but wanted to introduce you to the concept. So this is something that I've been learning more about recently. So this is solid state fermentation. So this is, you know, in some ways a very ancient process, right? So here, this is a modern version of an ancient process with a Koji type reactor. So solid state, broad field, numerous reactor designs. Like we could spend an entire presentation just on solid state fermentation reactors. And so the design really depends on the level of sterility you need, the manual labor input, so kind of the cost of manual labor that you're looking at, right? So some reactor designs are really intensive on manual labor, especially in countries where manual labor is cheap. And the product that you're trying to produce and slash the feedstock that you're growing the product on. So this kind of reactor is used to make sake, soji, miso, soy sauce. So using species like Aspergillus oryzae and Aspergillus soji. So you can learn more about that and Google it. It's kind of a fun topic. So apart from food processes, solid state fermentation is also being used to make leather. So this was kind of recently, for some reason, I'm not coming up on the slide. Hold on. There we go. So we're not distracted by the wave. So this is from Bold Threads, which is actually right across the street from ABPDU. This is from their website, so definitely check it out if you want to learn more, because they have some more information there. but you can see the kind of the process on the top. They're actually using mycelia to grow and growing it in a solid state fermentation to produce this kind of leather that you can see here. So they've partnered with a couple of companies, I believe Adidas, Lululemon, a few others, to actually make product lines using this to supplant traditional leather. And it's kind of ends up being more ecologically friendly to produce this faux leather in this way. And they have some kind of points about this versus synthetic leather, which I'm not an expert on that topic, but I just thought people may find that interesting, so they can dig in more if they want after the presentation. So let's look at two other bioreactor types. So this on the left, you have an Airlift bioreactor. So this was more popular, like maybe the mid to early 20th century up till the 1980s or 1990s. Still used today, so niche applications available is what I say here. And they're ideal for shear sensitive cells because the way that you're actually doing your mixing is through air. So you're pumping in sterile air, and you can kind of see in the diagram here how you're doing this. So you have your medium, and you have that flows through in a valve here, and then you have your compressed air coming through here. And so all the mixing done in the reactor, kind of both, we'll talk about this in some more detail, but basically both the mass and the momentum transfer that happens in the reactor is due to compressed air, which is kind of helpful, right? Because instead of having kind of separate systems for the mass momentum transfer, it's all wrapped into one. So the kind of problems with this and the reason why it's not as commonly used anymore is that you need conditions that are inhospitable for contaminants. So it's really hard to keep one of these systems totally sterile. So you can see an example of one to the right here. So these things are absolutely huge. This one, I believe, has been decommissioned. So these are kind of ideal if you're producing like an acid or producing an antibiotic or something that's going to be inhospitable for other organisms to grow in. So some pharma processes still use these, but almost all the processes that used to work through the Airlift bioreactor have kind of moved over to the continuous stirred bioreactor, which is on the right. So this is going to be the main focus of our talk. By far most widely used bioreactor design. CSTRs excel because of their sterilizability, excellent mass, momentum, and heat transfer, and low capex because they're widely adopted and standardized, right? So that's kind of a big part of the input here is that even if maybe the Airlift bioreactor would be ideal for your process, if you can make it work in a continuous stirred tank bioreactor, it may be preferable to go with that because the capital expenditure is going to be a lot less to actually buy a large scale continuous stirred reactor because they're commonly made. These designs are super standardized. You can play around with it. You can do some customization of the design, and all of that will just be cheaper than buying the same size Airlift bioreactor. And you also don't have to worry about actually pushing in, here you have 93,000 meters cubed per hour of sterile compressed air. So this is just to kind of get used, this is the plug for LBL and JBay and ABPDU. This is from the ABF, which is the Agile Biofoundry. This is the design, build, test, learn cycle. So this is kind of a methodology by which you would take your target molecule, so you have your putative target from the left, and how you would design a bioprocess and iterate on that design in order to produce that target in an economical way. So you start with your design and you have TA and LCA. So TA is techno-economic analysis and LCA is life cycle analysis. There's kind of an adjunct to design. So you have design in the sense of, okay, you have to come up with what are the enzymes that are going to produce this end product. how is it going to fit into biochemical pathways that are known? And then you need to actually build that design into the organism that you're using. So you could be using a standard organism like E. coli or Saccharomyces cerevisiae, or maybe you're going to want to use something that's more interesting like a Rhodosporidium toroides or Yarrow lipolytica. There's some interesting examples of yeast that I've seen used in ABF or filamentous fungi. Really kind of the gambit. So that's kind of what ABF is trying to do. Then once you've built it into your organism, you need to test it. So you can see the ABPDU fermenter actually there with scale up. So test includes scale up here. So test could be very small scale tests with even shake flasks at first, and then you would see how if you did get good results in a shake flask, you would move up to a larger scale to see if you were able to replicate those results. And this is a mass spec right here. That's how you test that you're actually producing the molecule of interest. And then, this is a brain here because this is machine. You'd use machine learning to take all the information from test and use it to iterate back into the design, build, test, learn cycle. So then you're improving your process over time and then hitting the target metric you want to achieve. So I wanted to show you all that because this is something that you'll probably hear here in biotech. You'll hear about DBTL and no one will explain it to you. So I wanted to actually give that explanation. And if you want to dig in more, there's some good resources online, and the one I've listed here, and I can point you to other ones if you do want to learn about this topic more. So this is where our group lives. Our group specifically lives more in the test scale-up level, but today we're also going to be talking about how that interfaces with learn. So I went ahead and put a rectangle around all of these. So it kind of segues nicely into what is the role of ABPDU. Again, I adapted this slide from JP shamelessly. He made a very nice slide and I wanted to use it. So this is kind of just shows the different scales that you go to, from research and development to scaling up a process to actually manufacturing a bioproduct at a commercial scale, right? So on the left-hand side, you have the micro, or I guess you could also say milliscale, because you're talking about milliliters, but you can even do microliters in a plate as well. So this is where you do your strain engineering, your strain testing, your medium testing, and something called oxygenography, which is trying to figure out basically what are the minimum components that your organism needs to survive and grow and thrive. And then your early process development, and I'll explain why you can only do early process development at this scale. People have been trying, this is actually a kind of interesting area in the sense that when I say early process development, some companies kind of argue that they can actually do a lot of their process development in the 96 well plate, which is pretty amazing when you think about it, right? So if you can do if you can use a multi-channel pipetter and you can use a robot in most cases, you can really do tons of fermentations and test a ton of different strains. And if you can actually simulate at least some of the elements of what it would be like to be in a commercial scale fermenter at that scale, and get even an idea of it, it will save you a lot of time and a lot of money, because then you don't need to, you can actually select the right candidates that you would then bring through the next steps of this process. So then you have bench scale. So this is JP listed 100 milliliters to 20 liters. You'll hear a lot of arguments about what that exact, liters to milliliters are there, but I think that's a pretty good summary, like pretty good range. So this is really where you do a lot of the process development. So you can develop the optimal pH, DO, and feeding profiles for product production and growth, optimize your medium, measure OUR and CER. I know it's not actually defined here. So OUR is oxygen uptake rate and CER is carbon evolution rate. And then you can determine the oxygen transfer rate that you're going to need. So I've introduced this here and we're going to go into a lot more detail. So if you're confused, don't worry, I'll explain. So this step is really one of my favorite parts, actually, is working in the process development bench scale level, because you can really this is where you actually start to do a lot of the engineering and understanding what your organism likes and what you need to do in order to get it to grow well. I know that you all have, I don't know if you've actually done the experiment or just seen it, but this is, the kind of stuff that you guys do in your lab, which I think is so cool for your class, is actually being able to see what the bioreactors look like and see what a process like that looks like. It's a really valuable experience. And big companies that are developing processes, they're doing the exact same thing. Like they're using the same kind of bench scale reactors to do process development. So then when you move on to pilot scale, this is, again, you'll get a lot of arguments about what exact volumes pilot scale entails versus bench scale. But 50 liters to 1,000 liters sounds about right. You could even have 1,000 liters could even be production scale for some things, but for industrial biotech, this is just pilot scale. So you're going to continue some process development, but hopefully your process is pretty well figured out from your bench scale experiments. And then I think this is the really key part. And if you listen to JP's talk, then you did learn about this. But you're integrating the unit operations and testing the DSP operations. So what does that mean? It means that you're actually getting to test to see whether you can separate your fermentation broth and isolate your product, right? Because what's the point if you can't actually get the final product in a form that's usable? So then the other step of this is you can produce your products for material testing and characterization. Kind of depends on what kind of data you want, how useful it is, but especially in an early stage for a company, this can be really useful because you can actually produce, on the scale of kilograms of your product, so you can do more product testing and purity testing. So also in the kind of scale up, but you're starting to move into manufacturing realm, you have demo scale. So we've moved on to meters cubed up from even talking about liters. So we have 10 meters cubed to 50 meters cubed. And we're talking, this is process validation, right? So you've developed your process in the bench scale, you've tested it in the pilot scale and it's working. You think you can scale it up to a larger scale. So you're validating that at the demo scale. And then that helps you develop the specifications for a dedicated plant or figure out what rental facility you'll be able to use for your process, right? Sometimes you'll probably be renting a facility for this. So depending on the scale that they're working on for demo, they may have the 10 to 50 meters cubed, but also have the commercial scale reactors at the same facility. So sometimes the demo scale to commercial scale is kind of a seamless transition. You're just testing at the demo scale. And so you also can generate market seeding material. So what he means by here is like you can actually produce a decent amount of material if you wanted to send it out to people who are interested in your product and have them test it and use it in their own processes and make sure it works. Like if you're producing a commodity chemical, like 1,4-butanediol would be an example. And you're going to have partners who are going to use it for synthesizing a polymer. Right? Like if you, on the demo scale, you can demonstrate that you can do the separation and produce a pure product and then give it to those partners and then they can produce their end product successfully. Then you have a lot more certainty that you're going to have clients when you actually move on to this next stage, which is the commercial scale. So economic production, and then there's still going to be some continual process improvement. And this is probably the biggest difference between pharma and industrial biotech is that in pharma, once your process is FDA approved, the goal is to not deviate from it in any way, right? Like you throw the batch away if there's any deviation, like even if it costs you $10 million, it's garbage at the point that you've messed up the process and there's any difference, right? Or in that sense as well, you can't really improve the process, even if you see, oh, well, this part of the process could actually do better if we change this variable. You just can't do that because you have your process FDA approved. So you do actually have continual process improvement that can be done at the commercial scale. And sometimes you're also linking back to this pilot and demo scale to test different changes and try them at the commercial scale, even once you've developed your process to that level. So this is a fun image from Genomatica, courtesy of them. So lab scale equipment is not equal industrial scale equipment. So this is funny because you have, you know, your Erlenmeyer flask and your glass beaker and I think you have a bioreactor on the left here, you have your volumetric flask. So you can't just use these things in a laboratory and say, okay, now imagine that those things are 100,000 times bigger and the process is going to run the exact same. Like, that's just not how it works. So that's kind of the point of this figure, and we'll go through how to think about that and how you can actually do small scale testing that's going to be able to work once you move up to a larger scale. So I have some core principles listed here for designing a fermentation process that's also going to be feasible at the industrial scale. So we have 3 kind of principles, and those are going to be, this is going to be our framing for the rest of the presentation. So the first one is using titer, rate, and yield. So these are commonly called TRI metrics to benchmark process development. So when you measure all 3 of these in the test phase of the design, build, test, learn cycle that you all now know about, it allows for a more nuanced comparison between competing strains, right? So if the most common of these people will look at is titer, right? Like, okay, how much am I producing per unit volume? But we'll talk about why that's important, but why you also need to think about rate and yield when trying to compare 2 different processes. So I have these listed out here so you can see what the units are for the TRI metrics. So titer, concentration, rate, product formation rate per volume, right? So grams of product per hour per liter. Yield is grams of product per gram of substrate, right? So mass of product produced per mass of limiting substrate. And that's usually going to be glucose. But it could also be fructose, it could also be xylose. It doesn't really matter for this context. Yield is going to be the same thing, because you can do it per gram. And I'll talk about a little bit like what is that there's actually going to be a theoretical yield. So if you've taken chemistry, then you'll know the term theoretical yield. So that same kind of term exists in biotechnology as well. So let's start with titer. So this is one that you may be familiar with. So why is it significant? So one of the main, it's other than the fact that it's cool, and this is how people usually benchmark, you know, what's the record for producing something, right? It's like, oh, I produced the highest titer ever. But obviously we're not just in it for bragging rights. We actually want to see what the economic consequences are and whether it makes our process viable. So it's significant primarily because it decreases the cost of downstream processing. And this can kind of come about in multiple different ways depending on the product. So it's kind of hard to define exactly what the change would be, but some examples would be for some reactions, like if you want to do a precipitation, then you need to reach a certain minimum concentration. It's different for each different product, but there's some minimum concentrations that you'll need to reach to actually do a successful precipitation and get a pure product, or mostly pure product that you can then finish your purification on. Another one that's kind of obvious because you've learned from JP about TFF and maybe from your class as well. So tangential flow filtration, where you're trying to concentrate, let's say a protein. The higher the titer that you have, if you think about it, then the lower amount of time you need to spend concentrating, right? If you're already at 5 grams per liter and you need to reach 10 grams per liter, and you really want to be at that 5 grams per liter instead of 2 grams per liter, because you're not going to have to spend nearly as much time actually processing your broth. I saw Doug's chat here. I like that. So challenges with getting a high titer here are product toxicity and enzyme inhibition. And these are kind of related challenges, right? So product toxicity is more like, okay, is your cell even going to be able to survive when you're at this at the level that you want to get to? That really becomes a problem with a lot of different compounds, like a fuel, for instance. This can be a big problem. So this is something you have to think about, and there are strategies to deal with that. And then enzyme inhibition, it can kind of be the same thing in the sense that, like, if you're inhibiting certain enzymes, then your cell may also die. But it's different in the sense that, like, let's say you want to, like, the enzyme that produces your final product. It can be the case sometimes that enzymes are actually regulated by their product, or that an enzyme earlier in the biochemical pathway is regulated by the product. So the enzyme becomes inhibited over time because as the product builds up, it actually slows down the flux into that pathway itself. So the strategies to improve titer here are really just addressing those challenges, right? So one, I don't know if you've learned about it in this class, but we can talk about it more in the question period if people are interested. You can use adaptive evolution. So you can do this through something called a chemostat or through continuous passage. And the continuous passage could be in bioreactors, or it could be in flasks, or it could be in 96-well plates. So basically a process where you're getting the organism used to being in a high concentration of the product that you want to produce. So you get, let's say you can chemically synthesize the product that you're trying to produce, or you produce enough of it already that you have enough sample. You could actually grow in a plate or bioreactor your organism with differing amounts of your product. And then over time, increase the amount of product that the organism is growing with. And basically what you'll do is, this is how evolution works in general, right? Is you're then selecting the populations that can continue to reproduce even in that context. So this can actually be a pretty effective strategy. Another one, so this can work if your product will phase separate between water and, for instance, like an organic layer. You can do an extractive fermentation. So there's different kind of ways of approaching this, but let's say you wanted to produce a fuel, then maybe you have some kind of organic overlay. Usually in research, you would use you'd pick an organic compound that wasn't your fuel. But if you're actually doing this at commercial scale, what you really want is you actually want the fuel or the chemical that you're going to use in the end being kind of the overlay. So being put into the fermentation reaction. So instead of just water, you actually have these two phases. And then as your organism produces the product, it phase separates to the organic phase. So then it's not building up to a level where it's causing toxicity or enzyme inhibition. Another one is transporter optimization. So what do I mean by that? So transporters are proteins that are on the membrane of a cell, right? So if you have transporters, if your product is extracellular, then if you improve the transporters for that particular product, then you can export them out more efficiently. So they're not building up inside of the cell when it's producing the product. And enzyme engineering, that's like a very broad topic that if you're interested in, I would encourage you to look into, but that's how you can kind of approach the enzyme inhibition question. And also just enzyme engineering, you could put as the strategy for all of these, right? Because you want the most efficient enzyme possible to make your product. So let's move on to rate. So rate, also commonly called volumetric productivity, is very important, mainly because it dictates bioreactor sizing and therefore a big part of your capital expenditure. So the amount of money that you spend to actually build the plant that you're going to be using, right? And it kind of makes sense if you think about it in terms of the term volumetric productivity. Why, if you have higher volumetric productivity, you just actually need less volume to produce the amount of material that you want to produce, that you need to produce to give to your partners or self. So what are the challenges with having a high productivity? So bottleneck enzymes, again, we could go back to the last slide, and enzyme inhibition, these are all kind of related concepts. an inefficient expression. So if you have a plasmid that you have in your bacteria or your yeast, and you don't have the perfect promoters and it's not being expressed as often as you want it to be, then you can play with that, right? So strategies, so this is just more related to growing the organism. So there's productivity in terms of the product, but you could also think of productivity in terms of cell growth. So identifying the limiting medium components can apply to both. Can apply to both cell growth and product formation, depending on the product. But it always applies to cell growth. So then measuring your product and byproduct formation rates at different growth rates. This is an interesting one because it's not as obvious, right? Like you generally just think, you want to grow your organism as fast as you possibly can. But what you'll find is that if you grow the organism at a different rate, then you may actually produce a product more or less. So this is just, there's not like a rule of thumb related use here. It's just kind of a suggestion that this can actually be a useful topic to explore in doing experiments and in doing this kind of process development. And this is kind of outside of the scope here, but I included it. It's like multi-omic experiments. So you're using genomics to look for mutations, because you actually do have evolution over time within a culture. Transcriptomics to see how much RNA you're making. So if you're actually like what kind of environmental effects are affecting how much RNA you're producing. And proteomics is like look at the proteins that your organism is producing. So you can actually utilize these to try to identify bottlenecks in productivity as well. So, and then yield. So this one is not as commonly, at least I've found, thought about, but it ends up being really important. And we're going to get into why it's so important in a couple of slides. So yield ultimately determines the economic viability of a bioprocess. So that's a pretty strong statement. You can make some arguments that like, okay, but what if you can't separate out if you have a great yield, but you can't separate out the product? Okay, well, it's probably not viable, right? So maybe that's not totally 100% true, but it ends up being true in most cases, and we'll talk about why. So the challenges with increasing your yield, redox balance, so oxygen uptake limitations, we'll talk about that in detail, byproduct formation, right? So if you're producing, let's say you have E. coli, and you want to produce a protein, and you're producing acetate, right? Every gram of acetate that you're producing, you're converting your substrate into something that you don't want. So you actually want to cut that off if you can. And then incomplete substrate conversion, this would be a really bad one too. So you finish your fermentation and you look at the concentration, you see that some of your carbon source or nitrogen source is actually unused. So you basically wasted that substrate, right? Because then when you go to the next step of the separation process, maybe you can separate and reuse it, but it's going to be really difficult to actually reuse in another fermentation. So you do want to use up all of your substrate if possible. So here's some strategies to improve yield. So optimize oxygen transport or oxygen uptake. So we'll talk about that. And fermentation optimization combined with pathway engineering. So that's a super general statement, but it really is an interdisciplinary process to improve this. So I'll just mention as well that, so as I talked about before, there's a theoretical yield. So in the same way that you can do with chemistry, where you can kind of look at stoichiometry of an equation and say, hey, this is the theoretical yield of this component. Like this is the absolute maximum amount. This is something I learned from Professor Corlett. I'm glad you mentioned him, this concept. So you have a given amount that would be your 100% yield, the absolute maximum that you can produce from all the reactants in the equation. You can do the exact same thing in biology. It just ends up being more complicated because you can't just take simple stoichiometry, because you also have to think about regenerating NAD and producing ATP as inputs to actually what your theoretical yield could be. So there's some different techniques out there, and if you guys are more interested in this, I don't want to take the time now, but you can ask questions later, and I could go into more about how people do that, how people make kind of genomic models and use that to determine the yield, the theoretical yield. Okay, so why is yield so important for the economic viability of a bioprocess? So this is a picture of our lab. You have a holding tank right here. You have the fermenter in the background, and you have a disc stack centrifuge right here. So this would be the first stage of separation in a process. Okay, so this is there's kind of an interesting paper that I found on this topic. So there's a link here if you want to check it out after as well. This kind of illustrates the more general concept well, I think. So I say yield should drive the cost of production. So the next slide explains the should a little bit more, but this slide, what we're seeing is ethanol production cost versus glucose, and that's your feedstock in this case, cost. And this provides a good example of the importance of yield, because if you look at the y-axis, this is the production cost of ethanol. And this little dotted line is basically where it's economical. So, and these different scenarios are 10 cents to 50 cents per kilogram, right? So as you see, as your feedstock cost starts to increase, you start to become economically unviable, right? And so this is related to yield, because yield is just the grams of your product that you're producing per gram of the feedstock, right? So I think that makes sense, right? So that's what's going to govern the production cost of your chemical, because it's really sensitive to the actual cost of the feedstock. Like if the glucose was free, then yield wouldn't be nearly as important, but it ends up being a big cost driver, and that's kind of more apparent in this next slide. So, and this is why yield should drive the production cost. So separation cost can become the main driver of production cost if titer is too low for an efficient separation. So there's two columns here, right? On the left-hand side, you have pessimistic conditions. So this is, you have a pretty poor separation. You see here that separation cost is actually the biggest part of the pie in this part, and it's still the highest percent component, even when we get to 2,000 cubic meters. But it starts to decrease in yield, starts to increase in importance as you scale up, even in the pessimistic condition. If you look at the optimistic condition, this is where your titer is sufficient for an efficient separation. And as we scale from 100 to 2,000 cubic meters, you can see that the feedstock cost starts to really dominate. There's a lot of different reasons for that, but you can just see here that like separation cost, it does play a big role, but that's why it's so important before you would ever actually deploy a process to figure the separation out and make sure it can be efficient. Like you would never, the pessimistic condition is kind of unrealistic in the sense that like that just, you'd probably never actually get to the plant scale of producing it if you couldn't have an efficient separation. So, and the separation cost, and I think JP talked about this in your last presentation as well, is that the separation cost and feedstock costs can also interact when cheaper feedstocks introduce contaminants that must be separated. So this is something, if you're a company looking at a process, looking at your fermentation process, you really need to have the fermentation engineers talking to the separations engineers, because let's say like you need to add a ton of anti-foam in order to make it so your process doesn't foam over. Well, that's going to be really hard to separate later, and it's going to be really expensive to separate later. So you actually do need to think about that early on in your process development, is making sure that all the components that you're actually putting into your medium are going to be separable from the final product that you want to produce, and not cost prohibitive to do so. So the next kind of organizing principle is that you can use mass, momentum, and heat transfer theory to basically define scale-independent bioprocess parameters to be used to predict bioprocess performance at scale. So that's a lot of words, but I'll explain it, because I know you're probably not. These are probably things that you all know, but I wanted to give you the kind of the formal terminology so you'd be aware, so you'd kind of be able to organize your thoughts within the formal terminology. So how do you generate results that will replicate at larger scales for an aerobic bioprocess? So today we're basically going to limit ourselves to aerobic because it's just going to be easier for the length of this presentation to limit ourselves to aerobic. And a lot of the same principles will apply to anaerobic, but obviously you can just take out all the oxygen transport quandaries that we're talking about. So this is a 300-liter fermenter at ABPDU. And then on your left, and then on your right, you have a farnesene production plant in Brotas, which is originally owned by Amyris, and then it was sold this last year. I'm totally blanking on the company that it was sold to. So I'll probably remember later and say. So how do you do this? So you constrain your pilot scale experiments by understanding what is feasible at larger scales, right? So I think this is kind of intuitive that it may be the case that at the pilot scale, or especially at the bench scale, something will work really well. But because there's a difference between those scales in terms of the geometry of the vessel, in terms of the agitation used, in terms of like the cost of what you could put into it, you really do need to think about when you're designing your pilot scale experiment, would this work in a large scale experiment? Like if you needed to add, let's say like you needed to add a solid, like a good example could be like if you wanted to add something that is basically like a bicarbonate or something like that to control pH, like a solid bicarbonate that wasn't totally soluble, right? Like add enough of it. Like, and then, okay, great. Like as your pH lowers and you're producing more CO2 off the bicarbonate, and let's say your pathway like CO2, like we could make that work at 300 liters. Like we could just like pour it in, basically. It wouldn't necessarily be a problem. But imagine trying to do that in a large scale facility. There just isn't the equipment to do it. So there's kind of different sides to this. There's like the common sense side of like, okay, like is this even practical? Is there even the equipment to do it? So it's one part of it that you need to think about. And then the other side to it is that just considering what parameters you can define and measure at the pilot scale or the bench scale that are then going to actually port over to a large scale. So let's talk about those. So I'm going to open it up to you guys. You can unmute your mics, or maybe we can do like a raised hand situation. I guess if everyone talks at the same time, that would be a problem. So what are the primary constraints on aerobic bioprocess at scale? Does anyone have any ideas or suggestions? Okay, you don't all need to talk at once though.
Is one of them that the toxic compounds build up more?
No, that's not one that I put here, but no, that's possible. I don't know that they would build up at like, if you're thinking about it in terms of concentration, the concentration is probably going to be the same, but I appreciate you giving it a shot to answer.
Nick Cap says in the chat that his group says oxygen.
Okay, well, that's number one. Cool. Okay, so awesome. So let's do this one. I mean, you guys can think about, there's one more. So there's two that I'm talking about here. So think about, so oxygen transfer rate is the first one. So this will give you a little bit of time to think about what the second one could be. And I'll check the chat as well. So oxygen is often the limiting substrate in aerobic fermentation. Scaling up the bioprocess based on maintaining a constant OTR is a strategy for ensuring that the bioprocess that works at pilot scale, and this also applies to bench scale, will behave similarly at the production scale. So we'll get into the details of what that actually means and how you can use that. Okay, in the chat or on here, any other ideas for the other one? And I'll give you a lot of props if you get this one, because I think this one is hard for students to guess. Okay, so that's totally fine. The other one, which I've personally found really confusing when I first got into this as well, so I'll forgive you for it, is heat transfer rate. So I think part of the reason why this is confusing is that if you work in a small scale bioreactor and you like feel the jacket on the reactor, right? The jacket is warm. Like you need to warm up the media, right, in order to get it running. But actually what you need to do when you're producing a ton of biomass and when you're producing your product is you need to cool the bioreactor. You know, I'll give some math to that and some reasons why, but it's basically the heat that's being produced by your bacteria or your fungi or whatever your organism is like the heat of actually their metabolism is very high. So if you want to maintain your culture at a given temperature, let's say like 30 or 37 degrees Celsius, which are common ones, then you actually need to cool your reactor. So this can end up being limiting and ends up being different between the small scale and the larger scale because of the way that heat transfer works, right? So heat transfer is limited by the difference in temperature between the cooling medium and the culture, or the reactor in this case. So, and the heat transfer surface area, right? So heat transfer from a jacketed vessel becomes less efficient at production scale. And the main reason why is because of the difference of geometry of the vessel, right? So as you get to a larger vessel, we'll just actually go back so you can see. So you'll notice here when you look, so you look at our fermenter and you look at kind of the diameter to the height ratio of the vessel, and you compare that. So this is one of the fermentation vessels, right? What do you notice is the big difference here, right? The height, this is really tall, and I mean it's more wide than this reactor, obviously, but the height to diameter ratio is a lot higher, right? So basically what that means is if you have a jacket going around this reactor versus this reactor, you're going to be interacting less with the actual heat that's coming off. Like you have less surface area that you're actually interacting with the medium that you need to cool. So we'll talk about how to deal with that problem and different designs for heat transfer that can help at scale. Thanks for playing along. I know it's hard to answer the question midway through. So how to maintain a constant OTR across scales. So these are some constants that you can think about, or some kind of quantities that you can measure that can be useful for this, right? So in the momentum transfer category, so it looks like I was playing in chat. So let's see what we have. Okay, so it looks like we're halfway. I think that'll work well. So the momentum transfer is basically mixing, right? So mixing and like agitation is what you think about. So the mixing time determines the time for the reactor to be perfectly mixed, right? So this ends up being really dramatically different at small scale and large scale. So a mixing time of 5 to 15 seconds is common between 10 to 100 liters, compared to 70 to 140 seconds at 100,000 liters. So, and I'll show you some cool figures later that actually demonstrate that. So that's one way to scale up. So you can be like, okay, I want the mixing time at my 300 liter vessel to be the same as the mixing time at the larger vessel. That could be a good way to think about scaling up your reactor and trying to make the environments you're growing your cells in similar. And then you also can use constant impeller tip velocity. So when you see someone say RPM for agitation, that's not really a way to scale something. It doesn't really help you in any way, because you actually need to know what kind of impeller you're using, and then what the diameter of the impeller is, essentially, right? So that, along with RPM, allows you to determine the impeller tip velocity. The reason why that's important is because the point of most shear on the culture will be at the end of the impeller tip, right? So by measuring the kind of maximum impeller tip velocity, you can try to understand what shear your culture will be experiencing in a 1 liter vessel, and it will actually scale over to even if you were looking at a 100,000 liter vessel. So This kind of interacts with the first one because it's helpful to determine what the maximum shear stress that your organism can deal with, right? Because in order to get the mixing time down as you scale up, you need to increase the impeller tip velocity. So this is something that you can test at small scale and will be useful, so you can actually define what would be possible at larger scale and then design your experiment based on that. Then for mass, and I have gas, because for this case, we're basically interested in gas, although you could also be pumping in a liquid as well. So you have for mass transfer, you can look at volumetric flow rate. So this is a really common one. You've probably heard the term VBM before. So this is easy to control. This is just literally the amount of gas flow to the amount of volume you have. And it doesn't require experimental determination. You just pick the number. And it is scalable. It will work in that way. But it doesn't account for different vessel geometries or sparger designs. So if you try to scale up your process based only on volumetric gas flow, you probably wouldn't get the same results as in small scale versus large scale. And then KLA, so this is kind of an interesting concept that I'll explain a little bit more in a second, but it's basically proportional to gas power over volume times superficial gas velocity. Don't really worry about that. It's basically a way to normalize how you can do mass flow in one bioreactor versus another. And you can experimentally determine that. And I'll show you how in this next slide. And that allows you, because you need to, the annoying part is you need to do it for each bioreactor. It's actually an experiment you need to do. But the great part is that it allows you to normalize for the geometry and sparger differences between reactors. Like you can actually use KLA and make a comparison between the 1 liter and the 100,000 liter reactor. So how can it be experimentally determined? Well, it's just some nice math here, right? So I'm not going to go through all of this because I know we're somewhat limited on time, but I'll give you all these slides so you can look at them. But basically, I just want you to focus on this. So the OTR equals KLA times concentration. Basically, this is the saturation limit for oxygen, and then the concentration that actually exists within the reactor. So basically, what you can do is you can do an experiment where you max out. So you get to the point where you have your organism uptaking oxygen, so taking in oxygen at the same rate that oxygen is being pumped in. And then you end up getting OUR equals KLA times the saturation, the maximum saturation concentration of oxygen times your actual concentration that you have. And this actually, because you can determine all of these at the same time, you have enough variables that you can divide OUR by these, and you actually get your experimentally determined KLA. The KLA is also going to be different depending on the medium. So that's another thing to consider, because the maximum OTR, OUR is going to be different in a different medium. But this is really useful, and this is probably one of the most used ways to actually port results that you're getting at the small scale to a larger scale reactor. And this is just a nice picture where you have air bubbles, you have oxygen inside of it, and then you can see just kind of the scale of the air bubble versus the scale of the cell. This is the case where oxygen transfer rate is greater than or equal to oxygen uptake rate. Just a nice picture. I'm not going to go through these, but this is all the things that determine KLA. So that's all. I'll give you the presentation. So if you are curious about this topic, you can look at it. There's a lot of different things that affect it. This tells you why you can't just use VBM to go from a 1 liter to a 100,000 liter reactor. Okay, so I'll try to get a little bit of participation here as well. So what if process OUR, so oxygen uptake rate, is too high for a given reactor, even with pressure, agitation, and aeration at maximum? So what do you do? How do you solve this problem? If it's like you've maxed out the pressure on the reactor, because that will increase that has to do with the saturation of oxygen you can possibly get. You're spinning as fast as you can, and you're putting in as much air as you can, what do you do? Looks like, can you differences between batches of media? So that's actually a good point. So that if you change the media, that will actually play a role here. I don't even think I have that listed here, so you get extra points because I don't even talk about that. Yeah, you could change the media, so especially if you're using anti-foam. Anti-foam is like the killer for OTR, just because of the way that it actually functions. It limits the ability for air bubbles to form. Like it kind of, like, why does it break up foam, right? Think about it for a second. Basically just like it limits the ability to form bubbles. So if you limit the ability to form bubbles, you'll break your oxygen bubbles more. And if you do that, you have poor oxygen transfer. So it's actually a good answer because I didn't even address that. So first option of two is you reduced your maximum OUR, but how? So you can reduce the maximum, you can reduce it by reducing your growth rate. And this can generally, the way people do this is by adjusting your feed rate. I'll chat again. I was kind of say starve, but then I like that, Doug. That was private to me, so I guess I shouldn't have shared it, but that's awesome. That's exactly what you should do. So you can see the figure on the right, and this is if you're more mathy, look at this equation, and this is just related to like kinetics here. So you have, this is how oxygen uptake rate is related to, so mu here is specific growth rate, and then X is your existing biomass, and you basically just have like, okay, how much oxygen are you using per biomass, right? So these kind of cancel out. So if you look at this, right, how do you lower OUR? Well, you either have less biomass. We don't want that. We want to produce a ton of biomass so we can actually do this process well. Or you lower the growth rate. Even if the biomass is at the same level, if it's growing slower, then your OUR will be lower. So this is from Tim Dodge. He is an engineer at DuPont, and he gave this presentation at the UCSD microbial fermentation workshop, which was great. If you ever get the chance to go to that workshop, I'd highly recommend it. So this is blue is batch growth. And this shows that kind of over time, you reach at, like, this is the maximum growth rate because you're not limited on your substrate, right? You're not limiting the amount that your organism is getting. So you max out and the OUR starts to climb up. But if you do a fed batch and you limit the amount that you're actually feeding the organism, you can actually keep the OUR low. And you could get, let's say this is your maximum point, right? You could then get to that maximum point a little bit later in the fermentation, and then you're not going to actually max out the OUR, and that can work better. So the other way to do this is by actual pathway engineering. So this is a paper you guys can check out if you're interested. But this is kind of like how Amyris approached using pathway engineering to reduce the moles of O2 needed per mole of product or biomass, because that's what was limiting their process. And the other way you can do it, this is kind of cheating. I wouldn't expect you to come up with this, right? It's like, okay, you said you maxed everything out. And I'm like, well, you can actually do other stuff, right? So you can use pure oxygen, but this is expensive and there's safety concerns, right? Oxygen, especially if you have any kind of ignition source nearby, oxygen is dangerous. You can reduce the culture temperature, and this can work, but then you have to use more power to cool the reactor. So that actually is not very practical most of the time, unless your culture is already running at a really high temperature. And then I guess I did say this, okay, limit anti-foam usage by testing different types to see which is most effective. This is like do a different batch of different kind of media, basically, right? So that's gonna really give you a big hit to your OTR. So I really like this figure. Whoever made this, it's like really beautiful. This is like, you know, like fluid dynamics type of thing. So this kind of, this on your left, you have a gas fraction, and you can see kind of the concentration gradients that exist in terms of, so they say gas here, I think they're specifically talking about oxygen, because that's nothing else would really make sense. So this kind of makes sense, right? So you have, this is your sparger. So these are where your air bubbles are coming out of. So this is like greenish to yellowish area. So you have pretty high concentration of gas. And then because you have, in your agitator region, you have red here, and that's where you have your highest gas fraction. And then you have these kind of dead zones, like down here, this is a dead zone. Like there's dead zones along here, and you can kind of see the way the gas is actually spreading out on the reactor. And then for glucose and B, they're feeding in glucose from the top, right? So and this is kind of the element of mixing time that I was talking about before. So if you have a 1 liter reactor and the mixing time is 5 seconds, it doesn't really matter where you feed the glucose into. It's going to be totally homogeneous in such a quick amount of time that it's not going to make any difference. But if you have a large scale bioreactor and the mixing time is more on the order of, tens of seconds or even minutes, if it's a 100,000 liter reactor, it's going to be like 70 seconds to 140 seconds is what we said. Then you're going to have very large concentration gradients, and you're going to have dead zones of glucose as well. So your organism may actually starve in those regions, right? This is another big difference between small scale and large scale. So why is this important? So there's two basic ways that you can kind of deal with this, because this can cause some issues. So you can reduce concentration gradients, and you can use impellers that result in better mixing. There's like Rushton are these impellers, and there's axial impellers. I think you guys have already learned about this before, which is why I didn't include that info. But the downside is that you're going to increase your power consumption in order to get better mixing. You can optimize the sparger design and or add multiple sparging entry points in the reactor. The downside of this is expensive, because you'll have to require custom equipment. You kind of take down the upside of having standardized equipment if you try to customize your 100,000 liter reactor. Another way is like the really simple way to do this is install a dip tube to add substrate. So you actually have the tube inside of the reactor. Let's say instead of dropping the glucose coming in here, you're actually dropping it in midway through the reactor. You're going to get much more even mixing if you do that. Downside there is same as a sparger design, that's like, okay, it's customized, so it's more CapEx. And then you have an increased risk of contamination if you have some kind of tube inside the reactor, right? So you just need to make sure that you're able to sterilize that tube, or at least like clean it in a really effective way. The other way to approach this is develop a strain that doesn't care about the concentration gradients. So resilience testing through pulse feeding. So you can kind of simulate these dead zones by, instead of feeding continuously into the reactor, you can just feed in, let's say like it's the mixing time of 60 seconds for the reactor. Then just feed in every 60 seconds and see what happens. Does it survive? Are there strains that produce about the same amount of product and some of them survive and some don't in those environments? That can be really helpful to actually figure out like which strain you want to use as your production strain. Then the other, so that was addressing kind of glucose concentration gradients, or more broadly, substrates like sugar concentration gradients. And the other one is you can do resilience through adaptive evolution and strain selection. And that kind of addresses both and I say facultative anaerobes are best. And the reason why is that if you have the dead zones, the gas dead zones here, and you have facultative anaerobe, then it's not going to die, even though it has no oxygen, right? It may change its metabolic state, so it may not be ideal, but it won't actually die. And then you can also you could do a similar type of thing. I haven't seen anyone do this, but theoretically it makes sense that like instead of pulse feeding, you could maybe pulse in air and like keep out air for a certain amount of time and see how your culture does. So there's kind of some creative ways that you can approach this is my point here. I want you to be aware of. Okay, so hold on. Why are you cooling the reactor? Don't we need to heat it up? I already talked about this a little bit, but this is just in more detail. So at first, obviously, you would need to heat up a medium in order to get to your optimal temperature. Let's just say it's 30 degrees here, 30 degrees Celsius. However, cooling is more important because microbes generate heat through their metabolism. And so this is a helpful figure here. So this ends up being really important. And that's also part of the Amherst paper I linked before, is that this ends up dominating, like, not only do you want to produce more yield per mole of oxygen, you just don't want to have to use as much oxygen, right? And part of the reason why you don't want to use as much oxygen is you have to do more cooling. So this is how much heat the organism is actually producing per mole of oxygen. So just basic physics here, you're gonna have to supply, you're gonna have to supply just as much energy to cool it down, and actually even more energy because your system's not gonna be 100% efficient. So then this is another part of maximum OUR. So maximum OUR can thus be used to determine maximum cooling capacity that you need. And this is another reason to try to have your OUR be lower if you are limited on cooling capacity. So the cooling capacity depends on the cooling system design. So I talked about before, it's the delta T between the medium and your cooling jacket or coils, and then the surface area. So this is the surface area component. So if you have internal coils versus a jacketed vessel, the coils are going to be much more efficient at cooling. Kind of obviously here, if you look at the figure, let's look at C, right? And with C, you have much more surface area that's actually being exposed to the culture than with A, which is a jacketed vessel, right? The problem with this ends up being that this can create a lot more contamination. So you have to balance those two kind of constraints. And then E here can actually is, I think the reason why they don't actually show either a jacket or internal coil. Now you can use E with either, and this actually ends up being very useful, is that when you're exchanging the cooling fluid, right, you actually exchange it outside. So you have like a pump and you have like a set of coils outside where you're doing the heat exchange. And so that's one factor. The other factor you can do is you can change the cooling medium. So if you're using water, that's going to be somewhat limiting. But if you use glycol, some percent of glycol, then you'll be able to cool a lot more efficiently. But then you have to spend money on the glycol and it's more viscous. There's kind of some problems associated with that too. But I wanted to give you kind of the why this is a problem in industrial processes and how you can address it. So I have here bioprocesses that can run at high temperature, and rule of thumb greater than 35 C, are generally cheaper than lower temperature fermentations, which usually have to use glycol-based cooling in order to cool them out, cool off another fermentation, which if your product is expensive, that's probably fine. And this is an example of a real high density cell reactor with an internal coil. This is kind of cool. So if you want to check this out more, go ahead after the lecture. All right, last of the three. So I'll sprint through this so we can actually finish on time here. So design a fermentation medium with industrial scale operations in mind. So this is before I was talking about the bicarbonate, right? Like you don't want to actually use that in your process. That would be silly. Like you're never going to do that at a large scale. It's totally impractical. So this is also, I borrowed, I guess that's an interesting term. I borrowed this figure from Tim Dodge as well. So I think it's really nice. So this is a typical microbial cell composition. So, and I'm saying here that it drives the medium requirements, and it's kind of obvious, right? Like you are what you eat. It's the same concept here. So he didn't actually specify which microbe this is being used, like which he's modeling off of, but this applies pretty generally. But you'd have to think about it if you had like I mentioned before, like Rhodosporidium tortuloides is an example of something where you'd have to actually measure this because it's called an oleaginous yeast. So it produces a lot more lipids than other kinds of yeast. So your lipid number here is probably going to be quite a bit higher than 13%. Usually most of the dry weight is protein. Next most is RNA, and then DNA. You have some carbohydrates, and you have some water, metal ions low, and you have this kind of breaks it a little bit more into which particular metal ions end up being important and end up being, most of these are essential. Okay, so I have here why use a defined medium, right? So what is the defined versus complex medium? I didn't actually define it here. You probably already know the answer. But complex medium, you don't know the exact elemental composition of the medium. Like you could measure it, but like that's like a yeast extract would be an example of this. With a defined medium, you get better experimental control and more reproducible data and more process understanding in comparison to a complex medium. I think this is kind of obvious why that would be, but I'll go into a little bit. Is that so? Batches of yeast extract will actually vary a decent amount, like a few percent, in terms of like what amino acids are present within the yeast extract, for instance. They'll vary a little bit in terms of the other nitrogen, like the ratio of nitrogen within the yeast extract. So it may be that you're using one batch and getting one set of results, and you start getting another set of results when you do the next batch. Doesn't mean complex media is not useful. It is, especially in the beginning parts of an experiment. But that's the reason why you lose a little bit of your experimental control when you don't know all of the medium components, and it doesn't allow you're not able to do the mass balance that I'm going to explain in a second as well. So you can use the principle of the conservation of mass, which is something you've probably all learned about, to create a material balance based on our starting components. So you can do and you can also do a carbon nitrogen balance, and you can quantify the relative product, byproduct, and biomass yield and detect off-target flux. So what does that mean? That means like, okay, how efficiently am I converting my substrate into the end product, into biomass, and then how much byproduct like acetate or ethanol am I producing that I don't want to produce? And you can keep track of this throughout the fermentation. On the right, you just have, this is just me, I put in these letters here, sigma, so sum. So the sum of the mass in minus the sum of the mass out equals the net mass. And the net mass is what's inside the fermenter. So if at all times, all the things that you're putting in are basically on a scale. And for air, you're just measuring based on like the amount of air going in and the composition of the air. And then you're also measuring all the mass going out. So those would be your samples. If you're doing a chemostat, then it would be some of the medium. And it would also be your gas. So you could measure that with like a gas analyzer, either a mass spec or like a BlueSense, which is a company that does like OUR and OTR kind of calculator that you can plug your gas flow into. You can basically take the mass in, the mass out, and you should just know exactly what's in here. Right. This is kind of the fun part, that you can actually just use kind of these basic chemical concepts for biotechnology as well in fermentation engineering. So using stoichiometry to understand the implications of mass balance. So you probably have all seen some version of this equation, right? So the general equation for cell growth from substrate, there's a carbon substrate here, oxygen and nitrogen, right? So you can use this, and you can like, okay, look at your particular substrate. If you have like glycerol, it's going to be different versus if you have glucose. So you plug in your particular substrate, or like lysine or something, and you plug in the amount, and you basically use this, and like nitrogen and oxygen, you can actually kind of solve this equation to figure out how much of your substrate is being converted into cell mass. And you measure the mass of the dry cell mass, and you can then use this equation. And after you've accounted for the cell growth, you can use the same type of approach to account for conversion of the substrate to our desired product and byproducts. So what's the big advantage of this, right? It seems just like a lot of work. One of the big things here is like, one, you get an idea of your actual yield, like how much of the substrate is being. So how much of your substrate is actually being converted. And then what the really useful thing here too is that you can see if you're missing something, right? So if the mass balance doesn't add up, then you have some kind of byproduct that you weren't measuring. So this can be really important to identify. So you can go back, the strain engineers can actually go back and redirect the metabolic flux away from that byproduct you don't want into the product of interest that you do want. So this, I think, the last slide. So just a few notes on industrial scale medium. So anti-foam agents reduce OTR and can inhibit cell growth and product formation. So the rule of thumb is you want to test different ones if you're a fermentation engineer working for a company. You don't want to just go with the first one that you have or the one that you have left over from your last project. Like, you actually do want to test it as you get deeper into the project. You want to keep chloride levels low. This is if you're working in a stainless steel reactor. Even though, and so then the reason why is because it can actually kind of chip away at the chromium / molybdenum layer that actually makes it stainless steel. So you can kind of get pitting and destroy the reactor over time. And then medium components that are cheap at lab scale can be cost prohibitive at industrial scale. So like cheap in the terms of like, okay, as a company, whatever, I'll buy some B vitamins and put them in. But that's not really practical at large scale, like to just throw the kitchen sink at it, right? Like try all the amino acids, try all the B vitamins. If you give all of those things, you may get a better result, but you're going to want to eventually figure out which ones you actually need. And you can do that through using design of experiments or one component at a time experiments to determine which components are necessary. And then when designing a medium, consider the components that will either be converted to biomass, product, bioproducts, or treated as waste in downstream processing. This is kind of the thinking with the end in mind concept here, right? Like, someone's going to have to deal with this eventually. So you want every single component of your medium to be absolutely necessary. Because if it's not necessary, you should be cutting it out, because otherwise it's like going to waste. And you don't, like, the point of using this technology versus other technologies out there to produce materials is that it's more ecologically friendly. So you really have to consider that. And that's the life cycle analysis side of things, where like you don't want to produce more waste products. And you can check this out in more detail later. I just gave you a production process for bioethanol. So you can just see, like, this is what a full production process looks like. And you'd be pretty different for like a protein, but we can go over that if you have questions about that or want to know like what a process flow would look like for a protein typically. So these are the key takeaways. These are just the big three. Use try. Mass momentum and heat transfer theory allows you to scale up and use parameters that will actually scale, and then design fermentation medium with industrial scale operations in mind. Okay, I went 3 minutes over, but I'm proud of myself for getting pretty close there. So now I have questions from anyone. I would love to take some questions.
Yes.
You can just go ahead.
Yeah, hi. Hello, doctor. Just want to say it's very interesting, and it's really opened up a lot of sort of ways to think about fermentation and microbes involved in. I just have a question that I don't think I've ever heard of, the carbon. Did you say the carbon evolution rate?
Mm-hmm. Yeah.
Earlier, when yeah. I just would you mind just explaining that a little bit? It just seemed it's probably very straightforward, and I'm just missing something, but.
No, it is. Yeah. So carbon evolution rate is just the rate at which CO2 is being evolved from the culture.
Okay.
So, and the reason, like, people sometimes don't even think about carbon evolution rate because it'll end up usually being roughly equal to oxygen uptake rate, right?
Okay.
So a lot of times it just kind of depends what type of system you're using. Some people are only measuring OUR and not even measuring CER, but measuring CER, it can be different, right? Because like sometimes you're actually fixing some of the oxygen. So it usually is different, but sometimes people will just approximate CER with OUR.
And so if there's a big difference, then something's not right.
Well, yeah, it's not necessarily that something's not right. It just tells you a little bit about the metabolic state of the culture. It looks like you're talking, but I can't hear you, unfortunately. It doesn't look like you're on mute, but something went wrong with your computer.
I think there is. I just got something telling me. I couldn't hear the last part that you were saying. I'm sorry.
Oh yeah, no worries. I think I was just saying that it doesn't mean something's wrong necessarily, that if OUR doesn't equal CER, but it tells you about the metabolic state of the culture, right? It tells you if like, is all the oxygen that's being, right, because what is OUR is just all the oxygen that you're pumping a certain amount of oxygen, and then any of the oxygen, like when you reach a certain saturation point for your culture, you're consuming all of the oxygen. So then if all of that oxygen was then being used in like, let's say like the Krebs cycle, right? Then you would basically get a stoichiometric amount of the oxygen uptake rate, and the CER would end up being the exact same. But that's not actually going to be the case. It just tells you a little bit about the metabolism of your organism, and it's a different conclusion for different organisms, so it's hard to give like a definitive answer.
Thank you very much.
Yeah, you're very welcome. Thanks for the question. All right, do we have any other questions?
I'll go ahead and jump in here, Ethan. On point as usual, excellent presentation. Thank you again. I have a kind of non-topical question. I'm just curious about the lab and how it's working right now. I know typically there are interns through the CCI and other programs, I think Blur, right, that are offered through DOE. I'm just curious what it's like in the lab, who's working there, what are you doing, what are the interns doing, and what's happening right now, as much as you can talk about it,
Yeah, no, I can totally talk about it. So I'm actually, you can't see because I have this background. I'm in my office in the lab right now. So I've been coming in most days into the laboratory, and we've started to really pick up our experimental workload and get back into the swing of things in the last couple of months. So we don't have any interns coming in right now. I think that for the spring, it's kind of up in the air whether people will be able to come in or not. It really just depends on the local conditions going on, right? So there's been a lot of uncertainty, but in terms of who's coming in, the main difference is that like our scientists who mostly are working behind their computers and directing the experiments, they come in a lot less regularly. So they do have a program where people, like someone comes in every week and they can come check out the lab and all that kind of stuff. But that's the major difference. But we have people who are at the process engineer level. Most folks are coming in pretty regularly, but we have to limit it to, I think we're up to like 8 people that can be in at any given time. We fortunately have a pretty big lab space, so it's possible to do social distancing while running several experiments at the same time. But we also, I'm not wearing a mask right now because I'm inside of my office, but outside of my office, in any kind of common area, you always have to be wearing a mask. So we're just taking all the safety precautions that we can. Yeah, definitely. I'm not sure what we'll be able to do with interns in this next semester, which is just, it sucks to have uncertainty, right? To definitely apply or still put in your application, because we'll bring people in if we can, obviously, but there's kind of that outside influence that we can't control. But I know that one of our PIs did have someone do like a virtual internship where they did techno-economic analysis. So I think that at our lab and at other labs at LBL, most people for this last semester, it was just like all virtual internships if people did them. And I think that for the spring, it's just up in the air whether we're able to bring anyone in, because we're just limited on the number of people who can be in the lab at any time. Yeah, thanks for the question though, Doug.
So I have a question. So you mentioned a number of like bioreactors. So what's like the major difference between like the bioreactors that you were saying are ancient versus the ones that are like
Sorry, Maggie. I heard. I think there's just some like maybe some noise in the background. I heard bioreactors, and that was about it.
yeah, so you mentioned like a number of bioreactors, and then I was wondering what's like the difference between the ones that you said were ancient versus the ones today, like most currently.
Yeah. Okay. That's a good question. It's like different types of bioreactors or bioprocesses, and like what's used today. So that's why like I talked about like the wave bioreactor versus the continuous stirred bioreactor, the Airlift bioreactor, the solid state fermenter, and I'll share my slides with Doug after this, so you guys can look at them too. And there's some links there as well. So the most commonly used by far is the continuous stirred bioreactor, like really by far, especially at the industrial scale. But solid state fermentation, I think, is still a very interesting field and a developing field as well. So in solid state fermentation, you're growing your organism. Well, commonly it's like a mold. So I talked about Aspergillus oryzae. So that's like really commonly used in like a lot of Japanese processes. Like they've been developing it for thousands of years, right? So maybe soy sauce is a good example. And with bolt threads, they're making kind of this leather with solid state fermentation. So I think that's actually a really like developing field that people are trying to get into right now. I think it really kind of like what reactors are used just depends on the industry you're in. So like food, you would use more solid state reactors, like chemicals, less so. The interesting kind of area of overlap would be like, because you're growing your organism on a feedstock, right, in solid state, and then the new wave of feedstocks for liquid fermentation is like you want to break down these waste feedstocks and then make them into sugars, like a sugar solution, basically, and feed them to your organism. So what's kind of an interesting application, there's a few papers on this, is like you need to deconstruct the biomass before you can break it down into the sugar components. So what would be really cool is if you have a solid state fermentation where you're producing a product that's useful, and then it's also deconstructing the biomass, and then you can use that biomass to then make sugar that goes into another fermentation. So it's not really your question, but I'm just wanting to like plug that's kind of like an interesting way that these don't have to be mutually exclusive, and there could actually be a process that uses all of these, or like industries that feed into each other. But the basic answer is continuous stirred bioreactors are still the most used, and the main reason is just because like they have really good mixing. You can do good mass like gas transport through sparging into the reactor. So they end up working well in the sense that you can have a very high density. So you can grow up a lot of cells and produce a lot of the product that you want to produce. But you have mixing with the agitators, so you can actually keep everything suspended and relatively homogeneous, right? That's like the big problem with a lot of the other designs, is that it's hard to maintain a homogeneous enough environment.
Okay, thank you.
Yeah, you're welcome. Thanks for the question.
Hi, I had a question. Earlier on one of your slides, you were showing the different stages of how basically the manufacturing gets to a manufacturing stage, which is the largest. And there you mentioned how pharma and industrial biotech was different, and how for pharma, basically things had to be FDA approved and they would not be able to be changed, and how oppositely industrial biotech can be changed. And I was just kind of wondering who makes the regulations, like what regulations are there for the industrial biotech in order to keep these products that are being manufactured safe?
That's a good question. That's definitely not my area of expertise. I think it would just, it depends though. So I think if it was a food product, you'd have USDA involved, right? So it's not like you can just like ignore potential safety concerns of like the downstream from an industrial fermentation. But I guess more the point that I'm saying is like, okay, like if you're making a commodity chemical that's going to be used to make a polymer, like it's, you're gonna, the thing that's important is that you hit this like percent purity, right? And if there's like, it this guy has to be like maybe like 99.5% pure. And if the impurities that you have are slightly different batch to batch, like very slightly different, it could make a difference, but it won't necessarily. But when you have a drug that you're going to give to people, any kind of impurity difference could cause a different reaction, right? So I think that there is just a different regulatory structure, like obviously between pharma and industrial biotech, but within industrial biotech, it really depends on the process that you're doing and whether a person is ever going to consume the thing that you're producing, right? Because there's a different kind of standard of safety that you'd want to consider. Like if we make a commodity chemical and it like has an impurity we didn't know about, like, okay, it really sucks for the company that then gets that product, and then let's say it's not in spec, and then it like messes up their process. They're not able to produce their polymer. That's it. I think that part of the trade-off is like that's a little bit more acceptable than something being dangerous if we were trying to make a drug, and then it had some other component. So I don't think I really answer your question just because this is not my subject area expertise, but that's my stab at it.
Okay, thank you for your insight, though.
Yeah, thanks for the question. I think I saw maybe Nick, I saw you're maybe raising your hand. If you had a question, please go ahead.
Well, actually, I do have a question. I was just waving. I saw Brandon, and I wanted to say hi to him. So some of the students in there. But you're talking about kind of using, since heat is such a problem with this, you know, why doesn't everyone use like a thermophile of something that would do it at a very high temperature? And then you make all these things and you really don't have to worry about the temperature exchange.
Yeah, so it's a great question. I think people have tried that. So I'm getting a little bit of reverb, so you may want to, yeah, there you go. So like there's some like species of Clostridium that I know people have tried for like ethanol production. So there's a lot of stuff around ethanol, just because of like, fuel ethanol, making that. So there's been some research in that area. I think it's mainly just to do with the thermophiles don't have very high growth rates or productivities, right? So it is a problem for sure, having to eliminate the heat. So that is an advantage. And you do see, like, to your point, right? You do see that sometimes you take enzymes from these thermophilic organisms and then you put them into the organism of interest, and you use that to actually maybe produce your product or as the end product. But I think my understanding is that the main problem is that they're harder to work with for genetic engineering as well. That's another. There's like okay, they don't grow as fast. We don't maybe. You're not as used to like handling them as a fermentation engineer. Like those are 1 and 2. And the part that I'm not an expert on, but this is my understanding from talking to strain engineers, is that if you're trying to engineer E. coli, it's pretty well known how to engineer it, right? There's thousands of papers. Someone has tried an approach that'll work. So it's not even necessarily that it couldn't be better to do that, to actually use a thermophilic organism. I think that there's an argument that it could be. I think it's just not low-hanging fruit. Yeah, thanks for the question. It's a really good question.
Hi, Ethan. Thanks for the talk.
Yeah, thanks for attending.
Yeah, I had a comment and maybe a question. I didn't qualify for the internship because you need to be full-time, but I'm in my final class. So because some classes are only offered in the spring and fall, I find that's probably common for people that maybe their last semester before they will want to enter the internship, they might not be full-time. So that's why I was interested in the spring, but that's why I couldn't do it. Because I'm just in that one class. I don't know if anyone else ran into that or not, but it's just for students at community colleges, right?
So there's two different internships that end up being roughly equivalent. So there's the community college internship, CCI, and there's also something called SULI, the student undergraduate lab internship, I think is what it stands for. They end up being almost, so I did this program, right? So like SULI and CCI are essentially identical programs. Like the way that the prompts are written for writing your research report are different, but the groups actually meet, at least at LBL, the groups meet together and they basically have the same exact program. So those are kind of two programs. Like SULI, you could apply to from undergraduate. So like if you were transferring, you could still do a SULI internship. I believe with the SULI internship as well, and definitely look this up because there's on the LBL website and there's like a, Department of Workforce Development website on this topic as well. I believe you can do it for like the year after you graduate. So I think that there's a little bit more flexibility with like post-graduation. I don't know that that's what the case with CCI, but it could be like, I think that even if you're the community college student, there's no reason why you can't apply to SULI. Like it doesn't work the other way. If you were, let's say you were undergraduate at Berkeley, you can't apply for CCI, but there's nothing stopping the person who is eligible for CCI applying for SULI. So I don't have an answer for like exactly how the qualifications would work for the internship, but it may be that you could actually still apply for SULI. And then if you do end up transferring, that's definitely an option. If you're not eligible for that internship, if it was a different time, this would be an easier suggestion of like, hey, just email us and we'll see if we have if we can, like the difference between SULI/CCI, there's like that, which is an internship. There's also being a student research assistant at the lab. So that's something where you get paid by the lab instead of having kind of a stipend that's separate from the lab. Like we pay in for part of it for SULI, but it's also like technically you're kind of an employee of workforce and development. And there's benefits to doing that program because you get to write a paper. I think it is better to do it that way at first, but especially if we're able to open up the lab to interns, like definitely keep that open as a possibility that we will need to hire student research assistants at some point, because our student research assistants have moved on to other positions just because we weren't able to bring them back into the lab. Like one of your like former classmates, Jenny, now has a job working at a company. She was a student research assistant with us before. And I basically told her, I'm like, I don't have any certainty about when we can bring you back in. So let's look at the different opportunities that are out there. And I'll give you referrals and references and try to see if there's something that is a good fit.
Great, thanks. And then I have one question. Some of the companies that have formed that use the services at ABPDU that are in the Bay Area, what are some of them that are your favorite or maybe that are hiring?
That's a good question. I don't think someone's asked that before. So it's kind of a variant. One of my colleagues, Akash, whenever someone leaves the lab, like moves on to the next job, he always asks, what was your favorite project and what was your least favorite project? So I don't know if I have a favorite. So we've worked with a pretty wide variety of companies. So if you look at our website, you can see a list of some of them, right? I don't know if I want to like say that I have a favorite. So my internship, I worked with a company called Bioproducts, which they're using cyanobacteria to make a bioproduct that could be used for cosmetics, I thought that project was pretty interesting because we don't work with cyanobacteria/algae-like projects very much. So that was kind of novel for us. But I think that I don't know. I don't have a great answer because I think that each project kind of brings a different set of challenges. And I really enjoy working with industry because we get to learn a lot. Like we get to provide insight to them as well, but just like seeing the way that they design their process and like doing the tech transfer, it's kind of a pleasure just because you get to see it the way that someone else thinks. There's some really, really smart, capable people who we've worked with from industry.
Cool, thanks. Doug just said in the chat that Bolt Threads is hiring, and you said they're across the street from you. Did they also use ABPDU to start their project?
Yeah, so we have worked with them before, and I've personally worked with them as well. Yeah, so and they have like multiple different projects that they've done over time, but I mainly mentioned them for just because I wanted to talk about solid state fermentation. So that was nothing really to do with our lab or anything that I want to take any credit for. But yeah, we have worked with them before. And like we've also like other big like Ginkgo Bioworks and other they're actually in Boston. I don't know if they have a location out here now. We've worked with them before. That's another one you may know. Perfect Day is another company in this area. I think I saw that they may have some open positions, but feel free. Doug has my email. Just reach out. Just shoot me an email, and I can try to give you some info. And if I have any leads, like I'm obviously not like trolling LinkedIn or all the different sites looking for a job, but I think that there are definitely some companies out there that are hiring. This is like, even though this is like, you know, a difficult time, I think that some companies are really trying to bring people on.
Thanks.
Yeah, you're very welcome.
I'll just say anecdotally, since I've known people at ABPDU, starting with Deepti and other folks going back a few years now, that ABPDU must be a good place to work because there's a lot of stability in the people that work there. They don't seem to want to move on, even though you're so qualified, you could easily go to other companies. So it must be a darn good place to work. I think that speaks a lot to the longevity of people staying there and stuff. I just had a question. It was in the chat. I don't know if you addressed it yet, but Nick Cap asked if a virtual tour would be possible sometime of ABPDU.
That's a good question. I haven't thought about that. Yeah, I don't see why not. I think I just need to invest in like a selfie stick because I think I mean, I'm pretty tall, so maybe that could work just with my arm out. But I think I'd be able to actually show you a lot more if I had a selfie stick. But yeah, I don't see any reason why not. I think we're actually like we're going to be doing. I don't know if you guys have seen this. If you've looked at like houses recently or anything. If you're weird like me, I can't afford a house, but I look at houses on Zillow sometimes because I'm curious. But some of the houses have 3D tours, so they're actually going to bring in some of those cameras into the lab to make a like 3D tour possible of the lab. So you may be able to check it out, and I'll send you guys a link when we actually do get that set up. But no, it's a good question. I'm going to meet with Deepti later today, actually, so I'll bring that up to her.
I mean, even I know you have some really, you mentioned some of the specialized equipment you have there, but one you didn't mention, you didn't have time to, but you have the Amber. Even having videos on certain pieces of equipment that you have and how you use them would be really cool, like little profiles and how different parts of the process happen at ABPDU. That would be maybe a longer term project, but yeah, if we'll follow up with you on that. It'd be great to have some kind of virtual tour.
Yeah, no, also, so JP actually just made a video. It's going to be released for public consumption on the Amber system and like our standard operating procedure for setting it up. So whenever he actually finishes that and publishes it, I'll share it with you. He may also just share it with you anyways. But I think that would be really cool to see. That system's pretty awesome. Cool. Well, it looks like we're running up against time. I unfortunately have another meeting that I need to jump off to, but I want to thank you all for coming and attending. I know the virtual meeting is a little bit challenging in comparison. As a speaker, this is my first time giving a presentation like this, and the hardest thing is like, usually people laugh when you say things and there's like some kind of interaction. I can see faces and all this kind of stuff. So I think just, realize we're all kind of trying to get through this process together. And even though it's, not easy in every way, I'm really glad that you guys took the time to come and hear me speak and be engaged and ask questions and try to participate.
Great, thanks again, Ethan. Ethan Oakson, everybody, thank you for attending, and I will follow up with getting the slides out to everyone, and you can link up with him on LinkedIn, and he'll have his contact info, I'm sure, in the slides. So yeah, please follow up with any questions you have. Thanks again, Ethan.
Yep, you're very welcome. Thank you all. Take care.